Okay, so like I said, exam one will be due on October 3. I will make sure to have the exam one out about 8 to 10 days early so you can all do in your own time. I also recognize that there are people who do stuff ahead of time, and I would like to give some credit for that. So there will be an early date of submission with bonus points. So if you are able to submit it in the middle of the week, which is not that bad because you do have 2 weekends to do the exam. So if you manage to do it by Wednesday of the prior week, which is probably going to be 30th September or something, you will be able to get 5 bonus points on the exam. And if you are doing on a regular date, you don't lose anything, but you won't be getting the bonus points. And like I mentioned to you, you also have to finish the oral exam, and you should be fine. You should be ready for everything by the end of this week because we would have covered all the content, which means that you can do it anytime starting next week. Okay. So that's about 2-3 weeks in ahead. And let's now go back to module 2, which is the carbohydrate structure and functions. Good question. So when do we do the oral exam? So I can try to, well, basically you will get a stagger, like a whole lot of dates and times to pick with. So you can pick any of the dates and time. Just you have to pick one time. Like it could be like, it might be Monday 4 to 4:15, and that's the time you like, you can pick that. Or it could be Tuesday 6 to 6:15. So there'll be a whole array of time, and the sooner you pick, the better choice you get. I mean, that's what it is. Okay. And yeah, I would love to work with you and make sure that each one of you are doing well. Why do I do it an oral? Why do I do an oral exam? Not the answer is not to panic you. The answer is that when you are learning module 1, sometimes, and module 2, sometimes, you know, the way you are studying, it would benefit from a little bit of guidance. Like I would say, hey, you know, maybe you need to pay more attention to this kind of thing or that kind of thing. And so that immensely benefits student for the rest of the time, and they start learning better. Okay, so that's the goal. Okay, so I'm going to now module 2. I hope you can all see the PowerPoint. You can see, but not this one, guys. Okay. Will we get study guide for the exam? I have to check. Everything is on Canvas still. I have gotten a lot of feedback on different things which I've incorporated. For example, whenever people told me, like someone told me, like you should have all the modules up on almost in the beginning. So I have actually done that. I'm trying to see where is the, doesn't have really a study guide per se, but I can give you like a brief set of points that will help you. I do want to tell you that out of all of these modules, module 1 is the nomenclature, 2 carbohydrates, 3 is lipids, 4 is protein. No, 4 is nucleic acids, and then 5 is proteins, and then 6 and 7 are the one there is lots of memorization. So for 6 and 7, I have this Quizlet kind of thing where you can just go and review terms and all, but I plan to expand it to other modules. So it'll give you a chance to review things. So thank you for bringing that to my attention, and I'm going to try to have it. Okay. So what are we going to learn in module 2? We are going to learn about carbohydrates. That is, what are the common carbohydrate molecules? What are their structures? What do they look like? Why are they important? And we're going to talk about isomerism. So that's what we had included a little bit in module 1, but it will be much better understood in this module. And like always, please feel free to ask any questions, and you know, you can put that in the chat and use the best, most comfortable way that you have for asking questions. Feel free to do that. Carbohydrates are really important, especially we know that we eat a lot of carbs, which includes sugars. And that because those sugars are present and the bacteria is present in the teeth, they can cause fermentation, and that makes lactic acid. That acid results in dissolution of the teeth, and it results in formation of dental caries. So we really need to make sure we learn about carbohydrates in detail because it's extremely relevant to the field that you're going to be in. So we want to understand what does it mean to, what do the carbohydrates look like? What are their structures? What are their functions? And how do we understand them better? So just going back to module one for a second, what did we learn in module one? In module one, we were learning about the nomenclature of organic compounds, and we learned that there are different ways of representing an organic compound. Okay, so these are the different ways of representing. So carbohydrates are nothing but a class of hydrocarbons that contain polyhydroxy. So let me just, elaborate on the term polyhydroxy, because we are using this term polyhydroxy. So hydroxy here means that OH. Poly means many. So when there are many OH groups, that is a typical signature of a carbohydrate. Okay, so that's what is the basic definition. Now, an example of carbohydrates is sugars, and this is an important reminder. So what is an important reminder? Let me circle that for you, just in case, for people who like to make any kind of notes, these are available, of course, but just saying, when they have an O's in the end, that is the carbohydrate. Okay, so example is, of course, the most common example is glucose. The moment you see glucose and it has the O's in the end, you do know that that is a sugar. So there are two different types of sugars. One is the aldose and other is the ketose. So I'm going to pause here for a minute and ask you, does this aldo and keto ring a bell? And if so, then please share with me, why does it ring a bell? Okay, yep. I think we have one of the answers. Does anyone else want to share anything? Someone is talking, but I cannot hear probably. So one of them is aldehyde, the other is ketone. Okay, so one of them is aldehyde, the other one is ketone. And these are the two types of sugars that we will be working with. What do the carbohydrates do? They help us meet energy needs, and they constitute 90% of the plant matter. So that's what we will be, as we look through the structures, watch out for those two words, aldehydes and ketones, and you will be able to recognize them. It's always so nice to relate, different kinds of areas. So for example, we learned this in module one, and now we are able to use that application in something else. So that's something that always close to my heart, like you're learning something and applying it in different places, okay? So carbohydrates, what does the word mean? You can see the word hydrate here. So carbohydrates are the hydrates of carbon containing what? Containing polyhydroxy group, that is aldehydes and ketones. Okay, let's try to understand this a little bit better. This seems a very, like lots of jargon. So we're going to make an effort to break that down. So like I said, hydrates is like water, right? So it is containing, I'm just going to say here, it's containing hydrogen and oxygen. This is the composition, and then there is carbon here. So these are the three things. And what do they contain? Polyhydroxy. So let us see it with our own eyes. We don't have to rely on this. We can just see there is a OH here. There is a OH here. Similarly, there is a OH here. There is a OH here. So you do recognize here that there is two hydroxy groups. So I know that you are familiar by saying OH is I'm sorry, OH is equal to alcohol, but that is when it comes in the end. Okay, when it comes in the beginning, when you're talking about hydroxy, when you're using that term in beginning, then you call the same OH group. I'll just say same OH group is called hydroxy. Okay, so we have to learn to recognize that. So prefix hydroxy, suffix alcohol, all is the suffix. Are there any questions so far about the definition of carbohydrates or polyhydroxy or any of the words which anything seems complicated at this point? Okay. Yeah, I'm hoping that people are going to keep their things muted if they are not talking or participating. So I really appreciate that. Let me, yeah, thank you so much. Okay, so now if you see here, this is a very simple aldehyde and this is a very simple ketone. So now let me again remind you, aldehyde comes in the end. You see the C double bond coming in the end, it comes with the H. So that becomes CHO. Okay, so let me just write that down. And I think we talked about that in that class as well. The CHO in here, and this is just CO. So that is the difference because it's coming in the middle and it is using 2 bonds. So it doesn't need a hydrogen. Okay. So this is the glyceraldehyde and this is dihydroxyacetone. So don't be too much worried about the names. We're not going to worry about names. Okay. We're going to only worry about the concept at this time. This is an aldose and this is a ketose. Why do we call it aldose? Ald for the aldehyde and O's for the sugar. And ket for the ketone and O's for the sugar. So we are just trying to make it sound according to the name. So that is why this little note here, do you remember aldehydes and ketones from week 1? And that's all we're going to talk about. We're not going to be worried about acids. That's for another time. We're not going to talk about amines another time, amides another time. Today is the turn for aldehydes and ketones only. And that's why we learned them in the beginning. Okay, so let's proceed and try to see what happens when there are lots of, yeah, sorry, go ahead.
I do, but it's a tutoring class. It's a tutoring for her class.
Is there a question you have, Lacey? Lacey, do you have a question for me?
No.
Okay, all right.
Sorry.
It's okay, it's all right. Okay, so monosaccharides. Monosaccharide is like mono is single, like single, and saccharide is sugar. You know saccharide because you might have seen those packets or pouches where sometimes they call by different names. So these are simple sugars. They have 3 to 7 carbon atoms, and they either have an aldose or a ketose group. So again, we're going to see what this definition actually means. So let's look at the nomenclature. Prefix. They are named by the number or they are named by commonly popular names. Why is that? We will see that soon. And the suffix is always os. Okay, so one thing that is really, really good in this module compared to module 1, we are not dealing with 10 different suffixes. We have learned them all. Now it's a matter of applying them. When it is a 3 carbon, we call it triose. When it is 4, we call it tetrose. 5 carbons, pentose. 6 is hexose, and 7 is heptose. So that is by number. So that means we have now talked about the first part. That is, let's see, by number. Now we are going to talk about popular names. Why do we need popular names? Let us see. We can solve this mystery by counting the number of carbons. 1, 2, 3, 4, 5, 6. That's 6 carbon. You will soon notice that each of these is 6 carbon. Okay, that's going to be a little bit tricky. All of these are 6 carbon. If I call each one of them hexose, how will I tell this one apart from this one? Okay, so you may ask me, why do I need to tell apart? Do you see the difference in structure between D-glucose and don't worry about the D. So glucose and mannose, do you see the difference in structure? And I'm going to just number them for a second so that you're aware that's, what is the difference in structure between glucose, mannose, galactose, and fructose? Like, can you, just first three, not the fructose. What is the difference between these three? Do you see the difference?
The HOs on the different side.
That is correct. So yes, so the HO is on different side. And you may say, does that make a difference? Absolutely. You know, it absolutely makes a difference in nature. These are two different things. Glucose is not same as mannose. You give glucose to the patients, right? You don't really give mannose to the patients, right? So, I mean, you're going to see that this difference does make a world of difference. So I'm just highlighting here what the difference between these two is. Okay, and then there is a difference here between this with this, and that is right here. So these small differences in structure makes them the exact same compound with structural differences. Okay, so that, and these are all aldehydes. You see the group in here? I'm going to label that in blue so that you can all see here. CHO, 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 all are aldehydes. Okay, and this one has ketone. I'm going to label that in brown. So I would just want you to appreciate that these are different molecules. Even though they look similar, they have the same number of carbon. And that brings us to the reason why they have popular names. Why they have popular names. Because we cannot call each of them hexose. We cannot, if we do that, we won't be able to distinguish. Okay. So now let's go to the next part. So if there is any question from this slide, you can write it down now in the chat or raise your hand. Okay, so now things get a little bit interesting. We are only working with monosaccharides at this time. We are not going to worry about this. Again, mono means one, saccharide means sugar, that one molecule of sugar. Or one, it could be even just the ring structure. So sometimes when we see things, we may think like, why are we making this complicated? The thing is that we are trying to understand, science is all about understanding the nature around us. In nature, this monosaccharide exists in two forms. It can be a long chain form or it can be a ring structure. So these are the two possible forms. And I know they don't look same, but trust me, they are same. Okay. And so there is long chain structure is called Fischer, and the ring structure is called Haworth projection. So these are the two different names for two of these structures. Okay. So when we said glucose, galactose, and fructose, that's the kind of things that we did in the last slide, right? We talked about this glucose, galactose, and fructose. So instead of existing in this long chain form, they could also be circular. That's also a possibility. And when that happens, look at this nice numbering pattern. It starts not at O, but right after O, and you're numbering it 1, 2, 3, 4, 5, 6. A word of caution, and I would like you to pay your 100% attention here. You know, there is something that you need to remember is that these corners are C's. We have talked about this before. We have talked like how when there are corners, we should not just assume there is nothing there. These are C's. That's what makes this same as the structure. CHOH, CHOH. So H is not shown here. It's all there. So all I'm trying to do is I'm trying to show you this is 1, 2, 3, 4, 5, and 6. So that's what is the 6 carbon
I have a question.
ring structure. Yes, Gina, go ahead.
So where it says the O in the middle, is it just not a corner or something? Is that why you didn't count it?
Very good question. Very nice question. Okay, so let me just answer that one. So you see this O? That O is right here. So thank you for asking that. That reminds me to tell you that the O is right here. Okay. And why are we not counting the O? So, let us say we only like, it's funny, but we always count only carbons. If you remember those structure when there was diethyl ether and we were saying methyl, ethyl ether and all that, we never count the O's. We are only talking about carbon chain and carbon numbering. If, of course, if there were more than one O, like if there were O and then another O and then another O, then we will need to worry about numbering them. But at this time, there is only one O, so we are not going to be worrying about numbering them. We know exactly where it is. It's a unique spot on there, and that's where it is. Okay. So yeah, so good question, and keep asking those questions.
I do have one more question.
Yes.
I'm sorry.
No, you're good.
Okay. So I'm kind of confused on why you counted the other one as a 6. I mean, as a 6th one. Is it because a line is going up?
That is also a great question. I will tell you here that if you remember, let me just bring this back here. This is carbon 1, which is right here, carbon 2, carbon 3. Okay, so it starts over here. Carbon 1, which is right here. So this we are going to count as number 5. 1, 2, 3, 4. And the way it turns, we are not able to show, this is 5 and 6. So we are accounting for all the 6 carbons. So it will be unjustified if we count here 6 carbon, but here we say, oh, the one that goes up, we will not count that. So we have to count 6. But the counting is a little bit different in chain versus the ring structure. In ring structure, you start here, you say 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. Let me just say our loyalty is only to carbon. So we cannot leave the carbon even if it is hanging up there. So we are loyal to carbon. We like carbon. Okay. So we are going to be only talking about carbons. So we have the 5th carbon here and the 6th one here. Okay. So in the beginning, sometimes when you read a new concept, it feels like that's not sitting well with you. If that happens, maybe I would encourage you to go through more reading, more number of times of reading, and it will help. But having those questions in mind, always a good idea. Okay, I'm going to give 3. I'm going to give like a 2-second pause just to take a screenshot. Okay, so yeah, keep asking those questions. If you have anything in mind later, you can do that. And let's just keep proceeding. I do want to tell you that there is a reason for this ring structure formation. And so the kind of person I am, I just need logic for everything. If I don't have the logic, for me, it's not like, oh, you told me the chain becomes ring and I have to just believe it. That doesn't sit well with me ever. So I'm used to of that questioning. So I just want to tell you that, there is a reason why the ring structure, or there is a way by the which ring structure is formed. And that's what is shown here. Not that you have to go into the details, but look at this, how this is formed. The CH2OH comes over here. And there is an exchange of electrons. These electrons go over there. And, these are donated here. And this O actually forms the double bond. You see 2 electrons and that forms a double bond. And this formed this, by double bond, I mean like O forms 1 bond over this side. And there is existing already 1 bond over here, which gives us this structure. The reason I did not tell this to you in the beginning, first of all, we are trying to understand the concept. Then we are trying to understand how this actually happened. Okay, so now the reason why we brought this up. Let us look at these 2 structures inside the green box. In the green box, what you see here is there are 2 different variations. In one of them, OH is towards the bottom, and in the other one, the OH is towards the top. Does this really matter? It does matter. In chemistry, any of the positioning of groups does matter. Okay. So you have to be careful, and we have to learn a little bit of name on that. Now, let's look at this carbon, which has this number 1 here, which is what we studied in the last slide. This carbon is a special carbon. Now pay attention to this one. This is something you need for an assignment. This is something you need the concept for exam. And so this is a very important, also a very easy concept. Okay. So when you have this carbon, which is attached to 2 oxygens, it is called an anomeric carbon. That is the name given to a carbon attached to 2 oxygen, anomeric carbon. It does not matter if the oxygen is here or it is on the bottom or the top. Now you're probably going to start looking at other carbon. Is this carbon attached to 2 oxygen? No, it is attached to 2 hydrogen, but not really 2 oxygen. Is this attached to 2 oxygen? No. Is this attached? No, neither this. A lot of times, students would think that this carbon on the top is attached to 2 oxygen. The reality is that it is not. It is just attached to 1 oxygen, which is this one. This is the oxygen it is attached to. However, this carbon, I'm going to show it to you in green, because sometimes, you are looking at things and they don't seem to make sense. Look at this. It is attached to this and this. So do you see that it is attached to 2 oxygen? And such a carbon which is attached to 2 oxygen is called an anomeric carbon. Okay. So now there is a name for this. We like to name everything. So this is called alpha glucose when it is opposite. And when it is together, it's called beta. And for those of you who look for tricks to remember things, here is the trick coming, okay? The trick here is that how I remember it, I say same side, I can make a beta with this. So when it is on the same side, I'll call it beta. The one that is not on the same side, I'll just call it alpha. So this is called alpha. Don't worry about the D yet. Alpha, and I know there is this name, but alpha glucose and beta glucose. We'll come to the names, okay? This is Howard projection, and so is this. The reason it's a Howard projection is it is a ring. I'm going to pause here and see if you guys have any questions for me on this slide.
I completely missed this whole page. I'm sorry. For you. I'll just go back to the recording and look at it again.
No, I'm going to actually pause for a second. Can I get a thumbs up on if you understood this, or if you want me to repeat this? If you want me to repeat this, then, okay. No, if you understood this, give a thumbs up. If I see there are low thumbs up, then I'll just explain it one more time.
I got a question about the one structure that you had up there.
Okay. I see only one thumbs up so far. So I think it's time to go back and review this. Okay. And I do appreciate all your honesty. This is not the class where you're going to be thinking, oh my God, what the instructor will think. Instructor will be happy. That's all I can tell you. I'm happy to explain this to you one more time. Okay. So let us look at this structure again. I just want to point out to you that we are looking at the same ring structure. However, we have managed to find that there is one carbon that is attached to 2 oxygens, which is indicated by the blue lines. Okay. So now this particular carbon is attached to 2 oxygen. None of the other carbons is attached to the 2 oxygen. This particular oxygen, which is on the bottom, looks different than the one that is on the top. So what I'm going to do here is I'm going to show you a trick to remember the names. What are the names? What are the names we're talking about is alpha and beta. So I want to tell you how we know the names, this alpha and beta, and we'll show that in pink color. So when these are on the same side, I think it's easy to form the beta. This is just the way I remember it. There is no logic to it. I don't think scientists thought of that. This is how I thought of it. Okay, how I can help you guys remember. And when there is only one, that is alpha. So if they're on the same side, you can form the B easily. So that is the beta glucose. And the one that is separate which is alone by itself, that is the alpha glucose. It's too far away from this. Does anyone have any questions about how the alpha and beta is named, or what is an anomeric carbon? If someone else is saying something, then I'm not hearing it. So, okay, for now we will proceed. But this is something that you will, I'm pretty sure you have questions. I just
Can
know
you
it
please
now.
repeat it one more time? I'm sorry. Please, Dr. M, please.
Yeah. So, yeah, I'm going to repeat it one more time. So the carbon that is attached to oxygen is called anomeric carbon, pointed by this arrow here, right? This carbon here, and you may say like, I don't see a carbon here. I do have to tell you that if there is a corner, then there is a carbon. Any corner is a carbon. So this carbon is attached to 2 oxygen. This carbon is also attached to 2 oxygen. How can we distinguish them? Because they are actually 2 different molecules with different properties. And the way we distinguish them is the one that are on the same side, we call that beta. And the one that is on the bottom and away from the other O, which is not next to its partner O, is alpha. So alpha and beta are 2 forms of glucose sugar. Okay.
So
Now
the big difference is just the location.
Exactly.
of-
Excellent, Crystal. Yes, the big difference is just the location, and that's what makes it different. On the bottom, it clearly says alpha glucose, O on opposite side. Beta glucose, O is on the same side. These are the 2 configurations. Again, why are we studying them? We are not making them up. This is how it exists in nature. Okay, now I have a little fun exercise for you, which we did in module 1. And that exercise is that if you put your hand on top of each other like this, they're not superimposable. But if you face them, then they are mirror images of each other. Okay. So surprisingly enough, our hand is not the only thing in nature that is like this. There are many, many things that can be looking just the same, but they're not really the same. Okay. And you might not have paid attention, but there are things in nature that could look left-handed or right-handed. And you can tell me like, how does it look left-handed to you? Because I can't see it. So let me show you. Oops. With the blue one, this is kind of going clockwise, you know, you guys, and this is going anti-clockwise. Does this happen? Yeah, these are like real pictures. This is not like made up pictures, computerized pictures, right? So it happens in the nature that things can be left-handed or right-handed or clockwise or anti-clockwise. It's entirely possible. And you can see you are yourself an example where your feet and hand, when they face each other, they look perfectly identical. You put on top of each other, not superimposable. What makes the difference? These lengths of these fingers and these thumbs protruding out. And you can understand if one of you is left-handed and or most of the people are right-handed, a right-handed person would never want to sit on this. And imagine the plight of a left-handed person who has to sit on that one, which is like mostly commonly happening, right? So all I'm saying is there are these differences. Why did we started talking about these? Is because that is what we are going to see happens in the sugars too. So this is highly relevant to what we are studying right now. Okay. So here is what it is. These hand, they look same, or feet look same, but they are actually not really the same. So these things that look the same but are not the same, these are called chiral objects. Okay. Chiral objects are non-superimposable objects. And then look at this wine glass or this ball or the bat. These are superimposable. You put a wine glass on the wine glass is like just go like right there. And I'm not really perfect in this, but I'm trying, you know, you guys. So I'm just putting this other glass on top of this. It's just going to fit in like that. That is non-chiral. It's also called achiral. Okay. Okay, let me think of some example. What can I give you to ask? Okay. What can be a good example? Okay, I'm
Just to
going
confirm, Dr. M, oh, sorry.
No, go ahead, go ahead. While I'm drawing, you go ahead and ask.
So just to confirm, how you say Carl?
Chiral, yeah.
That means that it is the same, right?
Non-chiral means that they are not superimposable. They're same, but they're not exactly, you cannot put them on top of each other and they will match up like your hands. You know, if you were going to put this hand over there, you can see that this thumb is going to stick out on that side.
Right.
Though in the mirror image, they look like, wow, this is identical, but in reality, they are not. Only in the mirror they are. So an object that cannot be superimposed on its mirror image is called chiral. Okay. So what I have for you is this nice water bottle, or I don't know, whatever you call that. This doesn't quite look like a good water bottle, but that's fine. We can live with that. Do you think this water bottle is going to be, more like this right side or like left side? What do you think?
Right side.
Right side. Anyone who thinks left side?
I think left.
You think left? Okay. I would give you an example of the left side. I have actually a fantastic example of the left side. Actually, I should not have drawn this one. I'm going to draw something really. You guys don't go to the lab enough. Let me see if any one of you knows what this is. Sorry, this is like pathetic, but it's something. What is this thing? Do you know what this is? I'm sorry, you can draw a better one. Pretty bad. What is this thing which is like goes straight and then?
There's something you use in the lab. I don't know what it's called
Is a
though.
beaker?
A beaker.
We're getting there, guys. We're getting there. And any other guesses? I know you guys can do it. You're very close. You're this close. Okay. Let me erase this one because it's so pathetic. Okay, I see some responses here. Oh, yes, flask. Yes, so this is a flask. Okay, great. So we have a flask here. Whether we put the flask on right side or left side, it's going to look the same. Okay. However, if I create a little nose on this flask, and I have this flask, and then I have another flask where I create a very similar flask, but the nozzle is, flask is not a good example because of the structure, but that's fine if for now. And in the mirror, it may look the same, but actually this is the thing that will stick out of the flask. Okay, so all I'm trying to say to you is your hand is going to give you the best example where it is mirror image of each other. It looks exactly the same, right and left. However, we know it is not the same. Luckily, we do not really have to use our brain for this. I'll tell you why. There is a very easy method to recognize chirality. So if you are afraid how I'm going to do this in molecule, please don't be, because here comes the trick. So what is the trick? Let's see the definition and everything will become clear. A carbon atom which is bound to 4 different groups is chiral. Okay, don't look at the rest of it. We'll come to that. If there is a carbon atom, it has 4 different groups. It is chiral. What does the 4 different group mean? Because I know this R1, R2 can throw people off completely. Okay, so let me just draw these for you and give you a nice example, which is easy to understand. So I'm just trying to draw the same thing. If I have an H here and I have an OH, I have a Cl here, and I have CH3 here. No, I don't want to confuse you. It's hypothetical. Let's make it That's a functional group. You have learned about it. Aldehyde. I know this doesn't really, it would be nicer if it's here, right? And Cl stands for chlorine. Cl doesn't stand for carbon here. A carbon having 4 different groups is called a chiral carbon. So there is no mirror images matching. That is just for our definition. That's for our understanding. But this is a chiral carbon. Now, if you ask me, I don't quite understand this. Does this and this look the same? I'm going to tell you one thing. Look carefully. This is R1. This here is R1. This is your R2. This is your R3. And this is your R4. So you know what is R? R is simply the names of each of these groups. R stands for of any group that's not carbon. So if they are all different, they're chiral. Now I'm going to make a very small change, okay? And then I need your help. So when I was away, let's say someone came and draw this. Can you tell me by just a Y or a yes on the chat or no for if you don't agree, is this chiral or not? And if you don't know, just say don't know. But say something, please, for God's sake. You know,
No.
otherwise I won't know. Huh?
I said no.
No? Okay.
Yes.
So we have one no vote. Like how many others? Are there any other votes for any other thing? Okay, I have one more no. It's okay to say yes. Don't walk under peer pressure. Great. Don't know is also a great answer. Anything is a good answer. Okay. No problem. No problem at all. Well, I'm celebrating. You know why? I didn't hear a single yes. Okay. Okay. So all I'm telling you is, and let me draw this one more time, even though this takes a little bit. So I'm just redrawing this exact same thing. I have a hydrogen on the top. I have an aldehyde on the bottom. I have something here and I have something over here. So let me say what that something is. So sorry, this doesn't look like exactly identical, but you know, you guys know that I'm trying to just make this identical. It's not that you cannot do these lectures by yourself. It just helps if someone explains it. Even when this was a completely online class, I was just running virtual hours and teaching it as if it's a virtual face-to-face. So I hope that people who are there and are able to attend are really paying attention. Okay, looks much better. Now I'm going to do a matching. So, okay, no, and actually, let me do this. I'm going to do a counting. 1, this is hydrogen. Is this same? No, this is 2. This is separate group. Is CHO same? No, this is a separate group. That is number 3. Is OH same? Oh, yeah. This and this are same. I see. So it has only 3 different functional groups. 3 different groups. What about on the bottom? This is different. This is different. This is different. They don't match each other. They're all different things. 4 different groups. Now I'm going to write this down because I'm done my analysis. A carbon bound to 4 different groups is chiral. Great. So I'm going to write here chiral. This is not 4. I will say achiral. If you have any questions about this, please don't hesitate to ask now. What I expect you to get from this side, I'm sorry, the spelling went a little bit wrong. If a carbon has 4 different groups, it is chiral. If it has 3 different groups, it is achiral. And it could be any group, like for example, I don't know. This could be just it could be CH3, CH2. It could be anything. It does not matter. 4 different groups, chiral, 4, less than 4 different groups, achiral. Can carbon has more than 4 different groups? No, we know it cannot have. When all the groups are different, it is chiral. Do you have any questions about this? Anything that do you want me to give you another example? Let me ask you another question. Okay, let me see. And let me see if you say yes or no or don't know. Okay. So we have, I'm trying to make this simple. So I'm just going to draw these stick figures. A very, very simple question here. And here and here. And then I'm just going to write down, okay? So I'm going to write something different. I'm going to make here COOH, that's one side. My second side is Cl, that's chlorine. My third side is OH, you know, that's hydroxy. And my last side is just H. So right now I'm not focusing on this side. I'm focusing on whatever this yellow box contains, sorry, blue box contains. This particular molecule, is this chiral or achiral? And you can write your answer, especially we're looking at this carbon. All you need to write is C or A or don't know. That's fine. If you don't know, just say don't know. And that's fine. We can address it in the end if that's still an issue. My friends, we need a little more responses in here, okay? Yeah, so all 4 different, does that mean it's chiral? Yes, absolutely right. It is chiral. Whenever all 4 are different, it is chiral, okay? And if I had 2 OH, then it will immediately become achiral. So that goes that if you were paying attention, you will be completely rewarded in assignment 2, and you will feel like, wow, this started off real easy for me because you were paying attention, right? Okay, so does this happen in nature? Absolutely. That's why we are studying. We are trying to here solve those mysteries, right? So if you look at this central carbon, it is attached to 4 different groups. And that is what makes it chiral. Asymmetric carbon is chiral. Okay, and here I draw a simplified structure. Okay, so now let us see something that is also, when we are talking about these, we want to name them. You know, we are always trying to name them. You know, we don't want to lose track of what is what. So in this side, the hydroxy is on the left side, and in this one, it is on the right side. So since it's on the left side, it's the L isomer. And now you may say, why is this not R isomer? You know, English was not the dominant language when things were being discovered and named. So you will find out the reason very soon. But hydroxy on left is L isomer. Hydroxy on right is D isomer. And we're going to revisit this. So don't think we're going to go this slide real quick and try to do this one, okay? So, oops, it did not clear. Oh, it probably is. Okay.
Dr. M, can you go back one more time, please?
In this one?
Yes, hold on one.
And I do want to tell you one more thing is L is for levo and D is for dextro. I'm thinking it's, I can't remember, is it Greek or Latin, something like that. Levo and dextro. And that's what their terminology is for left and right. And that's how we get this L and D forms. Okay. Any questions from this one? Okay. Let's read point number 3. When a molecule has one chiral center, and you will tell me like, what is a chiral center? So that carbon containing 4 different things is one chiral center. So this carbon has a special status. If it has 4 different things, we call it a chiral center. And if it has one chiral center, then it has two different forms, L form, D form. Let's read the name now. It will start making sense. L-Glyceraldehyde, D-Glyceraldehyde. And you may say, what is that word? Because I learned it a little bit different. So it's an older way of representing an aldehyde. But at this time, we benefit from this because it is aldehyde and D-aldehyde. And we know that sugars are all either aldehydes or ketones. It helps us just identify this. So this is a 3-carbon compound. Okay, is it okay to proceed to the next slide and show you 2 chiral centers?
You lost me. The lower one I understand, where you put left and right and
Okay,
but
for the top one, what did I say? I said this is called L-glyceraldehyde and this is called D. And all we need to learn here is about the fact that we call this L and we call this D. And both of these are aldehydes. Okay, so right now we are not going to worry about the name. How are we naming this and everything? What we are seeing is this is the L and D. Yes, any questions?
Yeah, I'm confused about the difference. So the L, so it's the same thing, right?
It
It's
is the
just-
same thing, but look at this.
Okay, gotcha.
This is on the left side. Try to draw this. So this is on the left side. This is on the right side. And you may think, is that just because of hydroxy? Why are you not looking at these other things? Why are you like so obsessed about hydroxy? I didn't name this, guys. This is how it is named. It's named based on the last hydroxy group. And you will see it in the other one. It will get, I know we'll go to 6-carbon, but trust me, it'll not be that complicated. It'll get easier. For now, for the definition's sake, we just know that if we are standing in front of this molecule, if on the left-hand side is the hydroxy, we call it LEVO or L. And if we are standing in front of the molecule and hydroxy is on the right side, we call it dextro or D. Is that okay?
Yeah, that makes sense. Left and right.
Okay, so now we are going to go to, so again, sometimes, when we are doing concept after concept, it may feel like, wow, that's a lot of stuff. So yeah, I really appreciate you guys focusing and listening. And I hope you are going to keep up your focus, okay? So just saying here that there is D and L form. If you see here, this is a D. You don't have to worry about the name at this time. I'm never going to ask you the name, memorize this name. I don't believe that that's going to be helpful here. So just look at this one and I'm going to tell you why it is D. I'm going to do it with a green box so it is easily understandable. Just look on this side. This is on the right-hand side, it is D. This is on the left-hand side, it is L. This is on the left-hand side, this is L. This is on the right-hand side, this is D. Same molecule, different names. Same molecule, different names. That's what makes it different. This has 2 chiral centers. Now you may say like, wow, this is getting a little bit complicated. Don't think too much. We're just going to focus on the bottom one. This carbon has attached to 4 different things. I hope you can visualize that. This carbon is attached to 4 different things. If they were attached to same thing, then H and H or OH and OH, you will see the same thing. They're all attached to different things. So this is 2 chiral centers. Okay. Because 2 chiral centers, this carbon is not, it has 2 hydrogens and an oxygen. So I mean, this is not a chiral center. Okay. Let me clear this. L and D forms. What are these L and D forms called? These L and D forms are called enantiomers. This is a special name. We learned about this in module 1, but here is for you to learn it, with examples. D rotates the light. When you put a plain polarized light that is not having all those, it's not a mixture, it's a plain polarized light. It rotates the plane of the polarized light to the, if you put a polarized light, it puts the direction of the plane light to the left for L and for right, D for the right. So that is why the name came You don't have to go into the details of this, but it's just for you to know that this molecule can rotate the plane of polarized light to the right if it is D, and if it is L, it can rotate it to left. You're not going to see any applications for this at all. Okay. So I do want to tell you that, these two are mirror images of each other, because if you put a mirror in between, it will just justify how these two look. These two are also mirror images of each other, but these two are not. Even though they are the same molecule, you can see that difference. Where is the difference? You may say, like, I don't see the difference really. Look at this and look at this, just a rearrangement. So this is enantiomer. Let me just write it down. Enantiomer. This is an enantiomer. And I'm just trying to make it, same color, enantiomers. These two green, they are not enantiomer. But, our lovely chemists, they try to find name for everything because they need the names for some other purposes. And these are called diastereomers. So what did you learn? You learned about two different types of concepts. You learned about enantiomers and you learned about diastereomers. Okay. Again, I would stress here not to worry about thinking, am I going to remember all this? You will remember all this, trust me. You just have to go over it one more time. You'll be fine. And maybe one more time.
Are you posting the recording today?
Yes, I will post it.
Thank you.
Okay, so now let's look at this. L-Glyceraldehyde and D-Glyceraldehyde. Now we're getting a little more comfortable with this. And the 4 carbon, we'd already talked about this. The L side is on the left side and the D is on the right side. So I actually try to show you like, this kind of thing. So you can just kind of correlate it. Like right is on the right side and left is on the left side. And that's what it is. Now you may say this is other OH. Are you going to worry about every OH? I would say the answer is absolutely no. You're just worried about the last chiral center. That's it. Last OH. Does it keep following through? Absolutely. Look at this. You have now graduated to the 5 carbon. And you might ask me, where are the 5 carbons? Let me show you. This is 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. 5 carbon, look at the last OH. Left side, that's L. Left side, that is L. Right side is O, is D. So all the time, the last chiral center on the left side is L and on the right side is D. Does this keep going? Absolutely, guys. Look at that. All on the right side in the 6. And then again, if you're questioning where is the 6th carbon, let's draw it together. 1, 2, 3, 4, 5, 6. 6 carbon for each one of them. Look at all these different names. And you may be thinking, why in the world are there so many names? They're all different, guys. They're all different. That's why we need all these common names. We have talose, galactose, glucose, allose, glucose, altrose, idose, mannose. I'm not going to ask you, draw the structure of galactose. No, that's not what we need to worry about. We are learning about just the D and L here, and that's all we need to worry about at this time. And that makes it the complete summary. So we started with 3 carbon. We have 4 carbon. This is your 5 carbons. This is your 6 carbon. Okay. Now I'm going to ask you a question. I'm wondering if I should ask, maybe not in this time, maybe next one. Okay, so let's look on the right side. This is aldo triose, aldo tetrose, aldo pentose, and aldo hexose. So again, a quick reminder. Let's pay attention to these groups. These are all aldehydes, right? Look at that. CHO, CHO. You can pick anywhere. I'm just randomly picking CHO. And we all know aldehydes. We have our knowledge now done from module 1. We know it. CHO is an aldehyde. Do we have a ketone tree like this? Yeah, absolutely. We have a ketone tree like this. It doesn't have too many things. It doesn't have so much variation. So that's the ketone tree. Where is the ketone? Let's try to identify the ketone. We love the functional groups. So right here, we have the ketones. And then we have the rest of them, and the names are given here, D and D-glucose or D-sucrose, there is D-cyclose, there is D-fructose, there is D-sorbose, and D-tagatose. So these are all the D forms. Okay, I'm going to pause for a second. Okay, how is it going so far? Is it going okay?
I'm
Okay.
sorry, I know I talk a lot out the entire class. I'm sorry, class.
No, guys, you can go ahead. Yeah, go ahead.
I did.
You are
I
the
did.
elected spokesperson.
I am so sorry. Okay. I asked a lot of questions. I'm hoping I'm helping somebody else too.
Yeah.
I missed the last thing because I had to take my 4-year-old to the bathroom. So I missed that, but I can go back to the recording.
I can definitely share with you. Wait, hold on. Give me 1 second.
Okay. Sorry.
Okay. All right. Okay. So now I'm going to go back there and okay, yeah, I have there are lots of nice reactions in here. Choose your favorite reaction so far. You can even choose like yes or no for like getting it or not really getting it, or like should I go slower or faster. I'll just wait for you guys, just chilling. Choose a reaction to tell like how you are feeling so far, so I get a sense of like, okay, and don't worry about others, you show your reaction. I care about each one of you in it doesn't have to be others. So please tell me, how is it going so far, guys? Come on. Okay, that's good to know. Yeah, you can use your words, put it in the chat. Anyone else? Any reactions? Okay. Okay. I like that. I think one of the most interesting comments I got on my, I think I can't remember whether it was on my evaluation or it was on my rate my professor, where it said like, I didn't used to like chemistry, but after this class, I think I like it a little bit. So I think that was a huge compliment, because I do want to make you all fall in love with science, right? Thank you, guys. Thank you. I appreciate your writing and all that. Okay. So we just summarized by saying that there is aldehyde-based sugars, which are all listed in this nice tree table. And then there is ketose-based sugars, which is all here. And how do we know that? We know that because we know that aldehyde comes on the top. And this is all the aldehydes, right? This aldehyde, this is aldehyde. These are all aldehyde sugars. And they are all, this ones are all the D sugars. And all the OH is on the right side. See that? The last OH, okay? Okay, and then we have the ketose, and that is in the middle. So we also identify that. There is a ketose in here, there's a ketose in here, and in here, all of that. And these are all the D sugars also. Just trying to stick to one kind. And similarly, you can draw for for L. Okay, so now let's just quickly do these terms. They came in the middle, we didn't do them. Why are we doing them? What happens is, like, in your field, you'll come across these terms,
Next one question.
Yes.
So you said the ketose is a D form. The aldehyde is what form?
D form.
Oh, it's a D form.
Yeah, all are D. And in this table, they are all D.
Okay, sorry.
Can I tell you why? Because in nature, all the sugars are in the D form. That's why. Okay, so if you see here, the alpha and beta form. If you remember, beta form is the one in which the O's are on the same side and alpha is on the opposite side. Glucose for glucose, fructose for fructose. Why do we have the term pyron and furon? Look, the pyron ring is the 6-membered ring and furon is the 5-membered ring. Why do we need to know this? Because I'll tell you, like, chemists will use different names and different things. So if you come across like alpha D glucopyranose, you shouldn't be like, what is that compound? You should immediately know that's alpha D glucose. That's a glucose, basically. It's not something funny. It's a really nice slide. Like, it tells you about, this is the anomeric configuration, alpha beta. This is the configuration of the sugar. This is D, not L. This is the common name prefix. This is the ring type, I'm sorry. And that is the O's, it's the reducing, or O side is non-reducing. We're not doing non-reducing at this time. So this is all the O's sugars. So this is what the nomenclature looks like. Let's do this. Guys, this one is something we can do together. So name for me, I know this is written in yellow. You can pick any one of them and tell me, or you can pick all of them and say, is one L, or let's do one first. We can do just one and be done. Is one L or D? That's all you have to say, L, D. That's all you have to pick. Okay, I see all so far all else. That's great. Good job, everyone. So this becomes, this is what it is. It's on the left side. So if you're standing in the middle, facing the C, this is your right hand, this is your left hand. It's on the left side, it is L. So we can just call it L. All right, so just go to the next one. So almost all sugars occurring in the nature are D enantiomers. There's no L sugar. It's not even there. Okay. So now, is there any application of this in the real life, or are we just like learning chemistry like nonstop? Well, thankfully, there is lots of application. Now, when we use these big words, we're going to jog our mind, remind our mind. So in a chemical reaction, when the anomeric carbon is the carbon that is attached to two oxygens. And the anomeric carbon has an OH group. When it has an OH group, it is considered a reducing sugar. So we will talk about the reducing sugar, okay? And, you know, there is, we are going to skip the bottom one. We're going to go to the next, so we'll come back to that. All the monosaccharides are reducing sugars, okay? So there can be two types of sugar. One is the reducing and the other is non-reducing. So the reducing sugars have a free aldehyde or a ketone group, and the non-reducing sugars, they don't have that free group, okay? You know, that is the difference. And we are going to revisit this concept in next Saturday as well, because I can understand this is going to be like a difficult concept, right? Okay, so reducing sugars have the capacity to reduce cupric ion of Benedict solution. What does that even mean? So let's take a look at the practical applications. I hope some of you have done biology 102, 102, or like a basic biology. In that you do find out like, is this a carbohydrate? Is this a protein? Like that kind of thing. So that's a Benedict's test. So imagine you have a test tube. You put some glucose in it. You are then, or a glucose solution in it, and then you are adding this Benedict solution. What happens is glucose has these, has the free OH group. So it's going to reduce the copper. By reducing, I mean like goes from +2 to +1. When it does that, it becomes red in color. You know, a copper will change color in different forms. It is different colors. Okay. Yeah. So yeah, it goes from this to high reducing sugar becomes like really, really red. So you can identify the reducing sugars in a chemical test. If it is a non-reducing sugar and you put that, then it's not going to be able to generate red color. So red color is indicative of copper getting reduced and producing that red. Sugar that can reduce that, they are sugar that are able to reduce the copper are called reducing sugar. I'm going to highlight that here.
Make sure you take a shower after. No, you said
Okay.
take a shower now.
Okay, so now I'm going to skip to the next topic, but before that, let's just come back to that. There are some of the examples here. There is some of the sugar names given here, and I know that you may not be familiar with this, so you will see in the coming slides some of the sugar. And we are going to, like I said, we're going to touch upon this topic of reducing and non-reducing and Benedict's solution one more time. At this time, the only thing you need to know is a reducing sugar will give a red color in test tube, and a non-reducing sugar will not give a red color, it will remain blue. Okay, the more the reducing it is, or the more the higher is the concentration of sugar, the more red it will be. If not too much sugar, it will be orange. Okay, so the sugars can undergo a lot of reactions. You see, they have all these hydroxy groups. There is an alcohol, there is a ketone sometimes, whatever that is. What happens in the body is like they can undergo phosphorylation. Oxidation of sugar can create carboxyl groups. So these are all the different things that can happen. Reduction of aldehydes or ketone groups of sugar can create sugar alcohols. So these are all these different things that can be created. They can even be addition of sulfates or amines. So sugars are pretty versatile. They're not like a glucose is a glucose is a glucose. It just undergoes a lot of different changes, different sugars. And that's for specific purposes. So this is not for you to really memorize per se, but understand that sugars have more role than just being left in L and D directions and such. They have important role in metabolism. Okay, so we are coming towards this. I believe this is not too many parts left now. So we are coming to a part where we are going to join two monosaccharides with a bond. So we got a glucose here and another glucose here when we joined them. And when we join them, a water molecule like goes away. But here this bond is formed. This bond is called Glycosidic Bond. And I know the name is a little complicated. So if you find anything that makes it easy to remember, share with us. But it's Glycosidic Bond. I know this comes from like glycogen, thinking of glycogen or glucose, and that's what this bond is. That's a primary structural linkage, whether you're talking about disaccharide or polysaccharide. So again, the word di means two and poly means many. So instead of one saccharide molecule in mono, you have two or three or four or more. They can combine in two different ways. You know, chemistry, the molecules really love to play with each other, literally. So when they combine with each other in different ways, they can produce different molecules. So this is maltose and this is cellobiose. Okay. Now, there can also be polysaccharides, which can be, so this, sorry, this is still a disaccharide. Disaccharide means two. They are double sugars connected by glycosidic bond. And this is sucrose. A sucrose is made up of glucose and fructose. Lactose is made up of glucose and galactose. And maltose is made up of glucose and glucose. So these are all the different forms. These are all the different variations. You can write down the formula for each and, practice if you wish to. You might have to do that in the assignment. Please don't shy away from writing these formulas. And, if you need some help, please do ask for help. And I can, you just actually literally have to make a hexagon and an O and then make all of this. So how does this actually make a disaccharide? Does this like undergo a magic? Not really. It's called dehydration synthesis. And I'm going to focus our attention on dehydration. I know hydration is like, how people say get hydrated, like get more water, right? So it's kind of, you can take the word from that. So hydration is water. Dehydration is removal of water. So it's literally that is. So you have two glucose, you remove the water, and then these kind of combine together and form a maltose. And what happens if you hydro, that is add water? And what is this word lysis? You know, the lysis word means to cut, lysis. Lyse it, cut it.
Dr. M, can you say that again? So glucose is with water, maltose is without water?
You can say that this particular group here, let me show you with an arrow. This particular, oh, sorry, the blue color doesn't really do any justice. Okay, so with the orange, this water, it kind of goes away here. You see this? So the water kind of comes out here, and that is the dehydration reaction. In case of hydrolysis, when you're adding water, what happens is that the molecule, the maltose molecule, or any other, for that matter, sucrose or anything, will break down into 2 monosaccharides. Okay. So removal of water will make a disaccharide. Addition of water will break the disaccharide back into monosaccharide. Why is this important? This is important because you will keep seeing this in next module and in next module and in next module. So really appreciate how the same concept will be used to make polymer in like every situation. Removal of water, make the polymer. Addition of water,
Awesome.
break the polymer.
I'm sorry. Maybe I just missed something. You lost me. I thought I was getting it with, but I'm not.
Look
I
at
hope
this
I'm not the
molecule.
only
These
one.
are the 2 glucose molecule.
Yes.
When you remove the water and keep it aside, then you are joining this O with this H, the black part, just join the black parts. So the molecule comes together. So this monosaccharide and this monosaccharide make a disaccharide. Now they're one molecule. First there was molecule 1, molecule 2, this is one molecule. What is the difference between these two? The water has come out. So this removal of water forces this oxygen to pair up with this hydrogen, this bond, and makes it a disaccharide. Whereas the addition of water will make it, will separate it out. You can see here the addition of water. By addition of water, I mean, well, here you're adding water. Once you add water, then this water kind of comes up over here and here, and it breaks them apart. So remove the water, you make it die. Add the water, you make it separated.
How early can you add the water to make it a hydro? Like, does it have to be there?
That's a great question. That's a great question. It can happen. I mean, you can do it in the lab, but it does happen biologically as well. Like, you know, in the plant or in the human body, when it needs more energy, it will break the maltose into glucose, you know. And, you know, there is, you will see with other things too. Whenever you need energy, you break it so that you can separately use the glucose. I don't know if you have ever heard about glycolysis, but then the glucose undergo glycolysis, and then it goes through the Krebs cycle and electron transport chain and produces energy, because the role of the glucose is to make energy. So if you do not need energy, you condense it and keep it like a storage. You need the energy, you break it and use it. So you are not really doing anything. I guess the body reactions and the enzymes and the cell is gauging the need and doing it. But I'm saying you can actually, yes, you can do it in a lab too. Okay. But all good questions, guys. Everyone who's asking questions. And I encourage others to not feel hesitant. Always make sure to ask questions. You know, you are here to learn. Even if, you know, I know a lot of people can be supported, you know, by the college financially, but you know, you're spending all this time, you know, you should definitely learn all the concept. I encourage you, at least in sciences, you don't understand something, you should just ask, bother people till you don't get it. So now we come to polysaccharides. There are 4 types of polysaccharides: cellulose, starch, glycogen, and chitin. So we have heard of cellulose. Cellulose is present in all these plants, right? Also the starch. We know that there's starchy foods. Potato is starchy, rice is starch. So does that mean they contain these chains of glucose? Absolutely. That's exactly what it means. They're different. Some of them, look at the cellulose and they're a little different. And that arrangement makes all the difference. So what is polysaccharide? Polysaccharides are polymeric carbohydrates. They're repeating units of monosaccharides or disaccharides connected by this, what we call a glycosidic bond. That's what we studied in the last one. We talked about glycosidic bond. See, we learned here. That's what we're using over here, the same terminology. So starch is dehydrating you. I don't think it really uses so much water. That's a really nice way to think about it. Starch is not really dehydrating you, but it will use water to break down. So yeah, it's a good way of thinking that it will need lots of water to break it down. Yes. So the structures can be linear or branch. So what does it mean? I'll explain to you in the very next slide. Oligo means when sometimes, people use these fancy words, oligosaccharide. What does that mean? It's just like 2 to 10 is oligo, and poly means like many, 10, 15, 20, 30, 40, 50, that's poly. So in nutshell, polysaccharides are the molecules in which animals or plants store excess energy or use it to build their cell walls or outer shells. I'm going to ask you a trick question. Where is glycogen present? Have you heard about like something which is rich in glycogen? I'm curious to see what you have to say. I've got some recently. Very good. Actually, it is present in liver. Yeah, I mean, if you were eating the liver of something, whatever, it is present in liver. But you've never heard like, oh, the potato is rich in glycogen. Not really. Potato is rich in starch. There's lots of cellulose in plants, right? And then chitin is like covering of those those crustaceans, the insects and all, like that have that crunchy covering. Sorry, there's for the lack of better words. Okay, so this is what it looks like. You can see that there is different things here, different structures in here. So let's take a look. You see that there's cellulose is straight, starch like has two forms, amylose and amylopectin is branched. Look at glycogen, it says so branched. It's really branched. Why? Because the branching is per 10 subunits. This is for less subunits. So these look different. This branches of, and you may say like, what did you exactly mean by branching? So look at this. This is like sugars. Okay. And then this sugar sometimes will form another kind of bond with another sugar and branch off in that direction. And so all these little beads are nothing but each one of them is a sugar molecule or a monosaccharide. So the plants keep making this, they work hard the whole day, they photosynthesize, blah, blah, and they keep storing the energy as starch because, they can't just keep storing this individual glucose molecules. Too much work. They need to store it in a condensed form, and that's when starch comes in handy. So here we go, just showing to you, just putting everything in perspective. A lot of structures are dependent on these molecules, right? If they form in a certain way, they form chitin, or they form cellulose, they form glycogen, they form starch. These are the complex or the polysaccharides. Yeah. Okay. So yeah, Gina, you can do whatever you need to do. Okay, so let's go to, we're almost done, guys. We're almost done. Like, I'm just letting you know that we are almost to the summary. So if you were paying attention, thank you so much for that. We right around the 4 o'clock mark, carbohydrates, there are all these different types. There is monosaccharides, there is disaccharides, there is oligosaccharides and polysaccharides. So in this unit, we were making an attempt to understand what is their structures, what are their functions? And we looked at one of the reactions. We looked at how do we identify them? So we can use a Benedict's reagent and identify if it's reducing or a non-reducing sugar. And we're going to talk a little more about it in our on our next class. But I just want to make sure that we have the whole thing wrapped around our head, even though if it's all the pieces are not perfect before you start the module next week. Okay, so yeah, for some reason, these numbers have become wacky. I'll fix them. Lots of uses of carbohydrates, especially for you guys. You are in this medical related field. We know glucose is used in restoring blood glucose level. There is fructose is used when, you know, how people who cannot be using glucose, fructose is used. Dextrose is dissolved in solutions and given intravenously to increase a person's blood sugar. There's maltose used in dialysis solution. There is dextrin, which is a polysaccharide of glucose, and it is used for used as an antithrombotic to reduce blood viscosity. So all of these have different roles to play. They even are a part of antibiotics. So there is this, I'm not sure if that's the right name, streptomycin. There is streptomycin is one of the molecules. And now that, my friends, you're really smart and you're identifying and working with all of these molecules, look at these structures. You know, there are this pyranose and, you know, sorry, this is not a pyranose. This is like more like a, actually that's a carbohydrate ring. And then this is the 6-membered ring. There's the 5-membered ring. All of these are carbohydrates, and you can see all of these structures in here. I would suggest that for the quiz, you should always take a look at this. There is no code. I think I removed the code, but you should definitely take a look at these words so that, you know, how sometimes people would wonder, what does the quiz entail? So, you know, if you know these terminologies, then you would be able to do well in quiz. For example, if you don't understand, like, for example, let's say, what is glycosidic bond? I would go back to that slide and see, oh, let me take another look. What is a glycosidic bond? Stuff like that. Okay, so with this, I'm going to stop recording.